So good morning, everybody, again. I'm so glad to be talking about a subject I'm very passionate about. A big part of my job is working in drug development, especially early phase drug development, bringing in new compounds, testing new drugs, because that's one and the only way out of the current paradigm or the current way of treating this subgroup of patients. And Dr. Starr nicely summarized how we go about current management, but he also told us about our challenges. This is one of those diseases where we haven't really made a whole lot of progress, and we still see patients, and we still see stats like 5-year survivals under 5%. And those are nothing to boast about. We haven't really moved the needle much in the last 5 years. There have been some sporadic improvements, and I'll touch about it, but there's a whole lot that needs to be done. And the only way we can do that is, one, advocacy, try to force scientific community, policymaker, industry, and let them know that there is this disease entity. It shouldn't be masked by any other cancers. You're all aware of this very fine actor who unfortunately passed away from high-grade neuroendocrine cancer, Irfan Khan. He was a big Bollywood actor, but also worked in a lot of really interesting and good Hollywood movies like Life of Pi. And so this disease affects everybody, rich or poor, young or old. I've lost patients, teenagers with high-grade neuroendocrine cancers. So we really need to come together and find better treatments, and the one way we do that is through clinical trials. And I'll go over some of the trials that are, of course, there's so many trials happening at any particular point of time, and I can't really go over everything, but at least some focal points so that I can give some ray of hope to our high-grade patients, their family members, that we are working towards it. We need to do better, but we are kind of getting there. For this particular talk, I would focus mostly on poorly differentiated neuroendocrine cancers, which are small cell cancers, large cell neuroendocrine cancers. We also have been educated by Dr. Hal Henderson that there is grade 3 well-differentiated NETs, which is also technically high-grade neuroendocrine cancer, but it's a little different disease than poorly differentiated neuroendocrine cancer and needs a nuanced approach. And fortunately, we also have now trials focusing on these grade 3s, high grade 2 well-differentiated NETs, which is a different biology. So I'm glad overall I think we are making progress, but a whole lot needs to be done. A lot of my interest in working on clinical trials and drug development for high grade really stemmed out from lung cancer. I work in a part of country, Appalachia, which unfortunately suffers from a very high incidence of lung cancer. It's a traditional tobacco growing belt, and a lot of tobacco use from early age, and we see a lot of lung cancer there. Unfortunately, we in Kentucky are among the top percentile for lung cancer incidence. And a big chunk of lung cancers, if you see, are type of neuroendocrine cancers. These could be small cell lung cancer, large cell neuroendocrine cancer, and carcinoids. Today, we will focus more on small cell and large cell because those are big subtypes of high-grade neuroendocrine cancers. Usually, 14% of all lung cancers are small cell lung cancer, but in Kentucky, 18% of all lung cancers are small cell lung cancer. That kind of gives you, a little glimpse of what we go through. I've personally treated close to 100 small cell lung cancer in the last 1 year. That's how big this problem is. And that's what really fueled my interest in exploring drugs and new drug development in high-grade neuroendocrine cancer, because traditionally, a lot of the drugs and our drug development has spilled over from small cell lung cancer world into other extrathoracic high-grade neuroendocrine cancer because of somewhat similar biology, not identical though, okay? There's a small study that we did just to kind of understand the burden of large cell neuroendocrine cancer, the thoracic large cell neuroendocrine cancer in Kentucky. And in last 10 years, the relative prevalence had doubled even for this such a minor subset called large cell. So this is a growing problem. Before we go further, you all are probably aware of platinum doublet, carboplatin etoposide, cisplatin etoposide. Those are our go-to drugs. No matter if you know somebody with high-grade neuroendocrine cancer, you could be in Brazil, you can be in India, China, US, everybody knows for metastatic disease, platinum doublet, right? And that really stems from work back in 1970s and 80s. So we really go back 4 decades, 5 decades, and we've been using the same drugs. They are very effective drugs. But that goes on to tell you that we really haven't made much improvement since our platinum doublet days. For second-line therapies, there is a slew of drugs. Some of them on-label, that means they have been FDA approved in small cell lung cancer. I've highlighted those, lurbinectedin being the recent example, topotecan. But then there are other drugs that have been recommended by NCCN and other consensus committees based on smaller studies, some promising data, and more importantly, lack of alternative options. But these are some of the agents that we often use in our practice because we really don't have good options, and there is some supportive data. And how we pick between these are primarily based on the availability, side effect profile, what patient would be able to tolerate. But our go-to is always clinical trial, because that's the best and scientific way to study a new drug and really advance the field where the next generation of patients can also benefit and really improve on our current standard, which is most often platinum doublet. In some cases, we can also use some alternatives. One of the big advances that we have seen in the recent past is use of genomics in high-grade neuroendocrine cancers. In cancers in general, we have access to technology. We can study the genome of tumor. You've probably heard this term called precision medicine, tailored medicine. Not everybody should get treated same way because everybody's cancer is very different, just like zebra stripes. And we know that for sure that everybody's cancer is very unique. This is more so for large cell neuroendocrine cancer. Not all large cell neuroendocrine cancer patients are same. Large cell neuroendocrine cancer of lung, pancreas, gallbladder, ovary, breast, they all are very different, both phenotypically, genotypically. They all respond differently to various chemotherapies, immunotherapies, other targeted therapies. So what we learned from this really pivotal work coming from Memorial Sloan Kettering is that when they sequenced the lung large cell, they could really put these large cell under 2 big, broad baskets. One is small cell-like, and another one is non-small cell-like. And just for simplicity, not going into too much of medical jargon, TP53 was one of the key mutations, and RB1, loss of that particular, would basically symbolize that it is a small cell-like phenotype of large cell. Because when we see a small cell lung cancer, the universal biomarker for that is loss of RB1 and TP53 mutations. But other 60% really don't carry that particular genetic fingerprinting. But out in real world, what do we do if we have large cell neuroendocrine cancer? Because of lack of better data, we all treat patients with platinum doublet as if they have small cell lung cancer. I'm not saying that's wrong, but that really comes from lack of data and lack of our understanding of the nuances and finer subtypes of this disease. So we are currently doing a small pilot clinical trial funded by a federal grant called K Awards. And we try to do upfront genetic sequencing and try to cohort patients. If it is a small cell-like cohort, we treat them like small cell lung cancer. If it's a non-small cell-like cohort, for example, if it's a pancreatic primary, should I do FOLFIRI-NOX? I use standard NCCN recommended adenocarcinoma regimen based on that particular site. It's a proof of concept study whether we can first find these 2 differences in a prospective study. Also, does the platinum sensitivity differs? Because in my anecdotal experience, small cell cohorts usually are very platinum sensitive, whereas non-small cell cohorts maybe not as platinum sensitive as small cell. So that's ongoing. The other work, because of our experience with small cell, really got me interested in studying immunotherapy. And fortunately, immunotherapy is that small advance that we have made, especially in small cell lung cancer. It's now FDA approved in frontline maintenance setting. But if you look at the data from small cell lung cancer study using drugs like atezolizumab or durvalumab, the small improvement that led to FDA approval was about 2 months, which is good, but not somewhere that we should boast about or stop the drug development. We need to keep going on, make these overall survivals much, much better. And that's where we got this idea about combining immunotherapy with a drug called rucaparib, which is a PARP inhibitor. So what we are trying to do in high-grade neuroendocrine cancer when we give platinum-based chemo, it's attacking their DNA and cause nicks in the DNA. And without the DNA, the cancer cell dies. Then why does the cancer cell survive and after 2 months, 4 months, 6 months, there's a relapse of disease? It's because the DNA has power to regenerate itself, to heal itself. And this particular drug, the PARP inhibitor, which is already approved for certain other cancers, so we know how it works, tries to slow down that process of repair. So it goes on very well with a DNA damaging agent. So in our particular trial, we start the patients on platinum-based chemo. We see patients who responded to platinum. Those patients are selected to participate in the study, and then we maintain them with immunotherapy plus a PARP inhibitor, which is a pill. I'm happy to announce you're probably the first one in the to know that we have finished the study, and we are actively analyzing the data and expecting to present it sometime next summer, either at ASCO or AACR. And why is this data important? Because if this works for small cell lung cancer, we might be able to test this hypothesis in extrathoracic high-grade neuroendocrine cancer, which is a critical unmet need. Small cell of prostate, small cell of pancreas. So we will keep you posted on how that's coming along. The other concept that I'm very excited about and might bear fruits in very near future is a concept of combination immunotherapy and an oncolytic virus. Now, viruses are bad things, right? We all know that. But we have used our understanding of bioengineering and converted some of these viruses to, instead of not harming us, but helping us, and they only attack cancer cells. One such virus is called Seneca Valley virus. This is some of the data on when Seneca Valley virus was injected in small cell preclinical models. You can see those big subcutaneous tumors. One injection of Seneca Valley virus and the tumor were gone. This data was coming from Dr. Charlie Luden's lab, who is a world-renowned small cell lung cancer expert in Memorial Sloan Kettering. So we know this particular agent works well in high grades. This also has activity in various other tumor types besides high grades, but my personal interest is high grades, so I kind of push the industry towards, hey, listen, you got to do the first trial in high grades. So we all got to do, right, our little bit of arm twisting there. This is data we just presented at NANETS a couple of weeks back. This is not from the preclinical NET model, but it's a pancreatic cancer model, just because it is very difficult to design a preclinical NET model which is also immunocompetent. But we wanted to showcase here, what we are trying to see here is when you add oncolytic virus like Seneca Valley virus and add immunotherapy to it, like Ipi, Nivo, you've heard those drugs, the immune doublet, those mice survived the longest. In fact, if you see those survival curves, majority of those the mice were cured 80 days and onwards. And at progression, the other interesting thing was wherever there are the star on the top purple line is that we were able to induce the remission again on reinjection. So this data looks very promising, and I'm very happy to announce that we've been working on this protocol for now close to 4 to 5 years, and we finally have an FDA approval on the initial draft of the protocol. And many thought leaders have been consulted with the industry, and we are expecting to roll out this study in 2025 net centers across the country so that everybody has access to this particular concept or early phase 1 study in near future. When would this launch? I'm thinking maybe first or second quarter of next year, but that is something that I cannot fully disclose. So those were some of the concepts that we were involved with and gets us excited and are going to kind of either we'll get to know some of the data soon or some of the trials would start. Now for the next 2 slides, I'll just focus on things that are happening right now that you guys can avail. And these are, I'm providing clinicaltrial.gov identifier numbers, so you can take pictures of this in case this trial is relevant to you. And the clinicaltrial.gov would then give you locations where these trials are open and the contact number. But out of all the high-grade trials, these are 6 or 7 trials that I'll go over that personally excites me. That doesn't mean that these are the best or the others are not. It is also no single provider, scientist, clinician can know about every single trial. So based on my understanding, these are my favorites, but please do not take that as a gospel truth. There could be other studies that I might have missed. Also follow your oncologist leads if he feels that there is some better trial for you locally or nationally. But these are some of those I would like to highlight. So one of the first trials coming from my good friend's lab, Dr. Anish Thomas, NIH. He's a small cell lung cancer expert, and he's been working heavily with DNA repair mechanism, especially ATR inhibitors. And this particular study is looking into combining the already approved drug called lurbinectedin, second, third line for small cell lung cancer, and adding ATR inhibitor, which is another DNA repair blocker called bezosertib. And this study is currently ongoing. I think this study might be limited to NIH for now, but please check on clinicaltrial.gov, might have additional locations. The other study I'd like to highlight is by our good NET specialist and colleague, Dr. Renuka Iyer. You might have heard about her. She's based off Roswell Park, and she is testing out this combination chemotherapy cocktail called you've probably heard about the drug FOLFIRI. It is a very similar cocktail. The advantage of this trial, or at least the theoretical advantage, is that the type of irinotecan we are using is packaged in a lipophilic sort of molecule, so it makes the absorption in the tumor better. So it is, in a way, a smarter irinotecan-based drug. Okay, so this study is ongoing. We are very close to opening it in Kentucky, but this study is also open at 4 other sites. So please check it out if this might be of interest to somebody you know with high-grade neuroendocrine cancers. The other study that really caught my attention, and that's specific for high-grade neuroendocrine cancer of cervical origin. Now, you probably think, hey, you're really going among the rarest of the rare, right? So from high-grade neuroendocrine cancer, we're going to just a gynecological high-grade neuroendocrine cancer. You'd be surprised there's a big community of patients who unfortunately suffer from small cell of cervix. And Dr. Fromowitz, he is exceptional and world leader in high-grade neuroendocrine cancers of gynonc malignancies based out of MD Anderson. And this particular study is also very interesting. So it is a drug called cadonilimab. It's an immune therapy doublet. So it's sort of like you consider Ipi-Nivo in just one drug. So it's a single Ipi-Nivo drug. It's FDA, not FDA approved, but it's approved in China for certain malignancies. But it's a very interesting drug. I think, and we know from SWOG DART 1609 that there could be role of CTLA4 PD-1 inhibitor, the Ipi-Nivo, based on a small study, needs to be further evaluated. But this one sort of combines the two drug in one drug. So I'm very curious to see how this study pans out, and if there's some success, this could be certainly investigated in other high-grade neuroendocrine cancer patients. Now, I would like to conclude my talk by a very, very exciting target in high grades, and it's called DLL3. This is up and coming, and I think has a huge, huge potential. So what is DLL3? You've probably heard of somatostatin receptors, heavily expressed in low-grade NETs, right? And we've used that property of NETs for both diagnosing and treating PRRT and other things. We were really lacking a good target in high grades. And DLL3, which is a notch pathway receptor, there are different types of DLL delta-like ligand, 1, 2, 3, 4, 5. So 3 seems to be heavily expressed in all high-grade neuroendocrine cancers, and actually even in grade 3 well-defined NETs, based on our work at Markey. So this is very well published, and a lot of different drug manufacturers are trying to target DLL3 with the help of chemotherapy, with the help of immunotherapy. There was a drug called rovalpatuzumab. The phase 1 trials of rovalpatuzumab, which was an antibody drug conjugate. That means one part of the drug attached to the DLL3 on the cancer cell, and the other part of the drug carried the chemotherapy moiety with it. And that was a smart way of giving the chemo directly in the tumor selectively. So the phase 1 data looked very promising. It was discussed in ASCO. Unfortunately, the phase 3 did not pan out. But that doesn't negate the role of DLL3. It just probably wasn't the right drug, or not combined, or the chemotherapy part of it wasn't right. So what really excites me targeting DLL3 is the combination with immunotherapy. And these are the 3 studies which I'd like to highlight, which are using or targeting DLL3 in high-grade neuroendocrine cancers. So the first study mentioned here is testing a very brand new compound, and these are called TriTox. Basically, what happens here is the drug, one arm of the drug attaches to DLL3, which is expressed in the tumor. The other arm of the drug attaches to T cells, which are immune cells. So it's really catching the immune cells, bringing it closer to the tumors. And the third arm, because it's TriTox, right? So tri has to have 3 arms. The third arm attaches to protein or albumin. And what's the role of that? We all have albumin in our blood and plasma. It tries to keep the drug around the tumor for longer. And that's the role of that third in a very, very simplistic way. So this study is currently enrolling in few centers. I think Harpoon Therapeutics is sponsor. So check it out if this is something accessible in your region, something which I found very, very interesting in our field. The other study is I actually recently sent a patient of mine to WashU, Dr. Tricolino's. I think his site has the study activated. Very similar concept. Instead of 3 arms, that particular drug is called BiTE antibody, so bispecific T cell engager. So one arm of the drug goes and attaches to the tumor, which has DLL3, all our high-grade cancers, and the other arm, so it's bispecific, right? So 2 attaches to the T cells. So it's bringing the T cell closer to the cancer, and that's basically the concept. So in this particular study, the patient gets pre-screened, so your biopsy tissue would be tested for DLL3, and if you have DLL3 positivity, then you might qualify for that particular study. Also open at various sites nationally, so you can look through clinicaltrial.gov and see if this might be relevant to you. The third study is using a BiTE antibody, very similar, but a different company product from Amgen. Again, same thing, DLL3 targeting BiTE antibody, and adding immunotherapy or immune checkpoint inhibitor sort of like Keytruda or nivolumab, similar class of drug called AMG 404. Now, this particular study, I have had some experience. We have it open in Kentucky, and I've treated some patients. And it's also a national, actually an international study at various sites have opened this study. So, but the issue with this particular study is only focused on small cell lung cancer. So if I have extrathoracic small cell cancer or high-grade cancer patients, which is outside lung, then I have to kind of channel them to another sites. But these are very, very interesting studies that I'm really keenly looking forward to see how they pan out in next year to two. But very exciting drug development happening targeting DLL3 with immunotherapy. And with that, I'd like to wrap up this small, brief talk. The goal of this talk wasn't really to go into brass tacks of all these clinical trials, but to sort of give a hope to our patients, our high-grade cancer patients, that you do have options, and please utilize clinicaltrials.gov and see if that trial might be relevant to you. There are more. I could not cover everything. And clinical trials is the only way out for us to really make a breakthrough here.